Hello, everyone. My name is Demetrius Jones, and I'm responsible for illustrating to you Figure 2 of the scientific research paper, which looks at CRISPR RNA, the complex, its interaction with Cas9, and its ability to cleave DNA and the plasmid DNA and oligonucleotide DNA, simulate what happens in real life with viral infections and plasmid, trying to get into bacteriophages, trying to get into other bacteria and infect them. So this figure looks at the two specific sites on the Cas9 protein, a complementary site and a non-complementary site. Cas9 protein that is used is from S. pyogenes. It is a highly conserved protein and sequence, and it is found in many other species and in many other kinds of bacteria. So Cas9, like I said earlier, has two nuclease domains. It has an HNH, an MCR-like domain, an RUVC-like domain. There is one HNH motif, and you can see as illustrated down by that schematic at the bottom of the page. It's actually part of the figure. I just thought it was very, very important and pertinent for you guys to kind of visualize what it is that we're exactly talking about here. So there's one HNH motif. This has been mapped out. And then there are three RUVC-like domains, and you can see them here. One here, one right before the HNH motif, and one shortly thereafter. And this is all on the Cas9 gene, protein, I'm sorry, of S. pyogenes. So some materials and methods for figure 2. So in order to first understand, the importance of both pieces of RNA, of CRISPR RNA and of transacting RNA and its association with Cas was kind of highlighted and outlined in figure 1. So now figure 2 is researchers are really trying to figure out which, they have these motifs and these domains mapped out. They just are not sure which interacts with the complementary, which interacts with the non-complementary. So to figure it out, they had to manipulate the amino acid sequence of the protein. And so alanine is used in both cases here as a point mutation. In the RUVC domain, aspartic acid is mutated to alanine, and in the HNH motif, on the complementary side, the histidine, position 840, is substituted for an alanine. Alanine is used in both cases and is commonly used in many other point mutations because of its side chain is closed, because the bonding angle, it's small. There's not going to be a lot of side chain interaction, and therefore researchers know they can remove the histidine or the aspartic acid, and you can put an alanine in there, and they don't have to worry too much about it reacting or denaturing or misfolding the protein. It's going to be kind of just a placeholder. And then scientists also radio-labeled the 5' end of the complementary or non-complementary end later, and then they will, based on the products of gel electrophoresis, you will be able to see some phospho imaging. We'll be able to see which side, complementary or non-complementary, is expressed depending upon which motif or domain has been mutated. And so the types of assays that we're going to do are an oligonucleotide DNA cleavage assay and a plasmid DNA cleavage assay. This is going to be after the Cas9 protein has been purified, and after the DNA and RNA structure has already been mapped out. Like I said, it's a pretty highly conserved protein. It's got a lot of common analogs in other species, and it's something they had a pretty good information about. So this is just quickly highlighting. This is in the supplemental index part of the study, and this is the amino acid sequence of Cas9. As you can see, here is the RUVC-like first domain. The second one is right here, just before the HNF motif, and the third one is over here, just past the HNF motif. If you look at the stars on specifically the first RUVC-like. You can see that D is where the alanine was substituted in for, the aspartic acid. And then this histidine right here on the HNH motif is what is being substituted for the alanine on the HNH motif. These are very highly conserved, like I've kind of said earlier. These are, they say they predicted, but they're obviously very knowledgeable about exactly where these are. A little bit just really quickly about the types of methods that were used for point mutations. They were generated with a quick change site-directed mutation kit, and then confirmed by DNA sequencing. The cleavage, the plasma DNA cleavage assay was imaged using a 1% agarose gel electrophoresis, and then visualized with ethidium bromide. And then for the DNA cleavage assay, cleavage products for the oligonucleotides you were resolved on a 12% denaturing polyacrylamide gel, 7 molar urea, and they were visualized by phosphoimaging. So these are the results. Like I said, you saw the schematic a couple slides ago, but this is the entire figure 2A. And if you look over here, you can see the scientists have drawn a schematic on the side there to show you where linear, nicked, and supercoiled folding is, so that you can see on the bands, depending on where they lined up, what type of folding is taking place. As you can see, there's a circular and a linearized. These are all protospacer 2 plasmid DNAs. So the wild, as you can see in both cases, the wild type is going to be unmutated. It's going to have both the CRISPR RNA, the transacting RNA, and the Cas9 protein. The Cas9 protein is going to be fully effective. So the products of these cleavages are going to be linear, as you can see on both in the circular and the linearized DNA. The products of wild type come out in the linear format. If you look at the mutation that has taken place on the aspartic acid, you can see when it is mutated, and so the RUVC-like domains are ineffective, you can see that the resulting DNA cleavage product has been nicked, which means it's open, which means that one of the strands is not being closed, and it's just kind of open to the environment. Same thing happens when you mutate the histidine that is on the HNH motif. The DNA product is nicked, and this kind of carries forth over to the linearized DNA. The mutation at the histidine on HNH and the aspartic acid on the RUVC domains and motifs result in nicked DNA products, unlike the wild type, which is going to cleave that DNA piece and then put everything back together. And you're not going to have open nucleic acids exposed to the external environment. So basically, the conclusion that they came to from this result was that there cannot be a mutation at either of these complementary or non-complementary domains. You need a fully functioning HNF motif and a fully functioning RUVC-like domain for proper endonucleolytic activity to take place. So this is figure 2B. Again, so this was looking at the complementary versus non-complementary strands. Basically, this was to figure out which motif or domain was responsible for binding with the complementary or binding with the non-complementary strands of the DNA. So as you can see here, we have a negative control on both sides. Then we have a wild type. And so the results of this figure were that in terms of complementary, you see the wild types. The wild type is the properly functioning protein complex, and it has this distinct band down here at 13, and the NT is nucleotides. And then you can see here that a mutation on the aspartic acid on the RUVC RUVC-like domain results in a very similar band. So that means that in this case, the RUVC endonuclease site is not it's not active, and in the complementary section it matches up with the wild type, which means that the HNH motif is the endonucleolytic site that is doing the work in this case. Another way you can confirm that is you look at the negative control, and that band lines up. It has a similar intensity to the mutation that corresponds to the histidine on the HNH, motif. So that means that when that HNH motif is mutated, it's not working properly, and that is confirmed by on the complementary side that the band of the negative control lines up with it. So for complementary strands, the HNH motif is what is primarily going to be doing the work, what is going to be doing the bonding and doing the endonuclease is going to be enacting on the complementary strand. Likewise, on the non-complementary strand, similar rationale. The negative control here at 50 nucleotides matches up with the mutation on aspartic acid. So basically, and then the wild type, which is doing its job properly, has a band that is very similar to the histidine over here. So basically, what it means is when this aspartic acid was mutated, this enzyme was not able to do its job, and it had a similar band to the negative control. Whereas the wild type down here at about 40 to 43 nucleotides matches up very nicely with the HNH motif. So when the HNH motif is mutated, the Cas9 protein is still able to carry out its job, which would indicate then that the mutation here had no effect on it. And therefore, you would say, or the researchers did conclude that the non-complementary strand, the endonuclease on that side, is dictated by the RUVC-like domain. So just some overarching conclusions. The Cas9 cRNA and transacting CRISPR RNA complex has two binding sites, which was illustrated in Figure 1, and both are very important. Both are required for good DNA cleavage, and then repair and closing of the double strand to take place. If only one is doing its job properly and the other one is inhibited or is ineffective, the DNA proteins, the DNA cleavage products are not going to be linear. They're not going to be sound. They're going to be nicked, and they're going to be incomplete. So the main conclusion again is that the HNH motif is responsible for the complementary strand of DNA binding, whereas the RUVC-like domain is responsible for the non-complementary strands, non-complementary strand binding. And as I illustrated earlier, the wild type combination of these three facets that together provide a critical immunity for bacteria. They produce linear plasma DNA DNA cleavage products, and any point mutation on either of these is going to result in a nicked product, and as both are required for proper functioning.